All right, it sounds like I have your attention, so we might just as well get going. We have Dr. Ignatius Tang. Do you ever call yourself Ignatius?
Sometimes I usually call myself Iggy.
In really formal occasions. So, Iggy, great to have you here today. Iggy is a postdoctoral research associate at the Systems Biology Initiative at UNSW. So let me scroll through this. He works under Sir Mark Wilkins and focuses on integrative data analysis, including large-scale biological omics data, genomics, omics, all omics?
Omics, proteomics, a little bit of metabolomics, but touch on that. But
yeah. And that playing with intracellular networks to understand cellular functions and how they are regulated.
Cool. Thank you.
Telling us about correlation profiling today.
Yes, I am. Lots of correlation metrics.
And the floor is yours.
Thank you. Thank you, Fabian, and thank you for the organizers. I'm Iggy. I'm going to talk about benchmarking protein correlation dataset against known references. And I work with Professor Mark Wilkins, and Loic is in the audience today. He's one of my collaborators. Very glad to have him here as a moral support and physical support as well. So I'm going to talk about what is a protein complex, what is protein correlation profiling, what are the correlation metrics used for scoring. Can you hear me okay? Yeah? All right, good. What is a high confidence reference protein complex, and how we can use randomization tests to benchmark PCP datasets against these known reference datasets. And I'll show you some example of significant matches. And I will talk about how we can use analysis of high confidence protein complexes to inform best practices, such as the choice of correlation metrics and how we can use it to better design our experiments. So most proteins do not function alone. They bind together to form protein complexes, which consist of 2 or more proteins, and they form molecular machine. And one example that people learn in high school is the hemoglobin. It is the protein complex responsible for delivering oxygen from your lung to the rest of your body. It consists of 2 chains, the alpha chain in red and the beta chain in blue. It's repeated to form a tetramer, 4 proteins binding together. It also has this heme group, which has an iron atom, and it binds to the oxygen, and bring oxygen to the rest of the body. One problem with measuring interaction is that interactions are measured one at a time, typically. So they tag one protein and they measure the interaction partners to that tag protein, and that needs to be repeated many times. So here comes protein correlation profiling. It measures many interactions at the same time. So we start off with mashing up the cells, and then it releases the protein complexes. And in this diagram, a circle represents a protein, several circles stuck together is a protein complex. And then we separate these protein complexes by size. So we separate them into 40 fractions or 40 bins. And typically the larger protein complexes tends to come out first. And then we measure each fraction using mass spectrometry. And from this we can see how much amount of each type of protein is present. And then we get a graph that looks like this, where the y-axis represents the protein abundance, and the x-axis represents the 40 fractions. And you can see in this diagram that the larger protein complexes come out first and the smaller ones come out later. And if you look at the actual data itself, it looks like this. On the left, you got the fatty acid synthase complex. It consists of 2 proteins, FAS1 and FAS2. And if you look at the abundance profiles, they look pretty much correlated. So it makes sense. They bind together, right? And then for the one on the right, it's a larger protein complex. It consists of about 12 proteins. You can see that it gets a bit noisier, but then there's still correlation between the proteins. So each line is a protein, of course. But there are challenges because machine learning strategies have been used to identify these protein complexes from PCP data, but this has been known to have high false positive rates for identifying novel complexes. There's known to be high false discovery rate of at least 85% in some studies. And for 2 independent screens looking for novel interactions in human data, there's less than 7% overlap. That's a bit worrying because these 2 studies, one from Cell, one from Nature, are very high impact, studies. So if they get it wrong, what can we do? So I thought I could take a step back and develop a method for benchmarking Saccharomyces cerevisiae Baker's yeast PCP datasets against high-quality sets of known protein complexes. And I'll use different correlation metrics to identify these protein complexes in Baker's yeast and in our dataset. So why do we use Saccharomyces cerevisiae Baker's yeast, which we use to ferment wine, bake bread, and you can buy them from the supermarket? It is because it is the most comprehensive interactome for any eukaryotic organism to date. It has the greatest number of proteins screened with respect to the size of the proteome, and we know most of the complexes already. So this is a typical analytical pipeline. We start with the PCP data, and we use peak picking to identify which are the peaks, and they correspond to different complexes usually. And then we use correlation metrics to calculate the correlation between each pair of protein or each pair of peaks. And then once we analyze many different correlation metrics, we use them as features in our machine learning. And then, for example, random forest. And once we got a correlation score for every pair of proteins, it comes up with a matrix. You can cluster that matrix to predict your protein complexes. And then we will compare it to known reference complexes to get our false discovery rate, etc. But today I'm only looking at 3 parts. We're focusing on the quality of the PCP data. We're looking at many different correlation metrics, and we will compare this with our protein complexes data set. So we use 2 types of chromatography. One is the size exclusion chromatography, SEC, which separate protein complexes by size. The other one is ion exchange chromatography, IEX, which separate protein complexes by charge, but there's no clear relationships between the size of the complex and the fraction number. So we use these are the data sets we have used. For the SEC dataset, we have 70 fractions and about 1,200 proteins, and it is our own dataset generated by Gene. And for the IEX dataset, we have 108 fractions and about 1,900 proteins, and it is a slightly larger dataset. And it is from a published data, which we have reanalyzed so that our data is comparable to theirs. So which protein-protein correlation scoring metric is better? So you have seen the fatty acid synthase complex. It has a Spearman correlation score of 0.96. For the other one, the 90S pre-ribosome, it has more proteins, right? So what we do is we calculate a mean correlation score per complex. So we looked at the correlation score for every pair of proteins, and then we get the mean of those to get the mean correlation score per protein complex. You can see that the Spearman correlation is 0.87, so it's a bit noisier. For the third one, the nucleoporin, it has a Spearman score of 0.5. You can start to see there's extra peaks in there, and it's a bit more noisier again. And the last one, it has a Spearman correlation of 0.37, and again, it's noisier, so it gets worse off scores. So, but which score is better? So in this study, I've used 16 metrics. They're roughly divided into 4 groups. I won't go into each metric because it will take ages. But there's the correlation scores, and there's the usual ones like Pearson correlation, Spearman candle correlation. You got the distances. Euclidean distance is the profile distance between 2 points, and people have learned them in high school. We use mutual information-based metrics. We use this because it can find more complicated correlations patterns, such as parabola, sine waves, ellipses. But we don't know whether these correlation metrics will work for our data set, so we are trying these. And we got peak and scores based on peak location and width as well. For our reference protein complexes dataset, we use the benchtop dataset. I won't go into detail here, but we have 518 complexes and about 1,900 proteins. They have found the consensus between 3 datasets, so it's high quality. And this dataset only consists of the high confidence protein complexes known to date, and many complexes are not included. So just look at give a first glance of our data. We took our SEC dataset and we calculate the correlation score between every pair of proteins, and we separate it into 2 groups. One is pairs of proteins that are known in the reference protein complexes, and the second set is the pair of proteins that are not in reference protein complexes. And if we plot the density of the 2 sets, this is what you see. You see that those in reference complexes have high correlation score, meaning that there's an enrichment for strong interactions. But if you look at the actual number of pairs, those in reference complexes are relatively small. It only covers 0.5% of the total number of pairs. So in our benchmarking data, we're really only looking at a small subset of our data, and the remaining protein-protein pairs that are not in the reference protein complexes are likely to contain more interactions. So to compare our PCP data to our known complexes, I use the randomization algorithm, and I'll go through this. So we start off with our abundance profiles. We calculate the correlation between every pair of proteins, and this gives us a matrix. And we compare our PCP data with our reference protein complex. So we have 5 proteins in this reference complex, and 3 of them were found in our dataset. We calculate the correlation score between every pair of protein, and we calculate the mean out of these 3 pairs, which is a score of 0.6. To get a background distribution, a null distribution to compare with, we randomly sampled 5 proteins. 3 of them are in our dataset. And we get the score for every randomized pair, we get the mean, and we compared the observed mean with the randomized mean. We do that 1,000 times, and if 999 times out of 1,000, the observed mean is greater, then we say that is a significant result. And of course, we need to adjust for false discovery rate because we are measuring many complexes, but I won't go into detail here today. But we are really focusing on stringency. We are only focusing on the really high confident protein complexes, which is a reduced subset of all known protein complexes. We are using false discovery rate of 0.05 for our randomization. And there are other methods that could detect protein complexes with higher sensitivity, such as using hierarchical clustering on our scoring matrix and using complete linkage algorithm, and that would predict most proteins to be in a protein complex, but we don't know whether it will be very accurate. So this is a glance of my result. On the left, you got your IEX data. On the right, you got your SEC data. The y-axis gives you the number of significant complexes, and the x-axis is the 16 correlation matrix. For the SEC dataset, Pearson correlation worked the best. For the IEX dataset, Spearman correlation worked the best. And then Euclidean distance did not find any complexes to be significant at all, which is interesting because a lot of people still use Euclidean distance to analyze PCP data. Mutual information type metrics such as MIC were not the best. Distance correlation, which is a strong contender to be a general correlation metric that can be used in many different applications, worked well in SEC, but not IEX. So usually when I compare many correlation metrics, I'll draw many Venn diagrams, but there's just way too many to be drawn. So I've represented the data in a heat map instead. Here I compare correlation metric A, which is the rows, and correlation metric B, which is the columns. And you can see that Spearman, Pearson, and Candle correlation metrics consistently identified the same protein complexes as significant. And of these metrics, there are few complexes that were identified as significant by one metric but not the other. And these ones, such as Euclidean distance and the mutual information-based metrics, didn't perform very well. And when we look at the IEX dataset, there are more correlation metrics that showed consistent results. This could be because the IEX dataset is larger, or it could be IEX data is easier to interpret. But yeah, we can use all these correlation metrics, but we still have to go back to the data to see what they mean, which peaks are more important. For this graph, we see a lot of proteins overlap here, so I think this peak is more important. But what happens if you have more complicated profiles? Is this peak going to be more important, or this peak, or this peak, or this peak? We don't know. So that's why we use peak picking, and Gaussian peak picking is one method that we can use. I've used the Prince software this time to perform the peak picking. And say if you have a protein on the top here, you do some smoothing, and once you fit the Gaussians, you found 3 peaks. And this is how peak picking works. And to leverage the peaks, I've used the co-apex score, also available in the Prince pipeline and developed by them. It compares the centroid of each peak and also compares the width of each peak. And it uses a Euclidean-like distance to get a score of 0.25 for this example. But it doesn't take into account of the peak height. So this is the result comparing before peak picking and after peak picking. On the left, you got the results for before peak picking. On the right, you got results for after peak picking. I also included results for including the first 4 fractions and excluding the first 4 fractions. That is because the first 4 fractions is known to interfere with the SEC data. So when we look at the results, peak picking reduced the number of complexes found for SEC, but not for IEX. Removing the first 4 fractions improved the SEC co-apex score. That is because there's lots of complexes coming out in the first 4 fractions, and removing those fractions help clean the data up a little bit. Even though peak picking didn't identify so many complexes, it does help with the interpretation of results, especially for SEC profiles. So when we use Venn diagram to compare Spearman correlation before peak picking and co-apex score after peak picking, this is what I found. What I see is that peak picking resulted in the identification of additional protein complexes. So that's a good thing. And I'll show you some example here. We are looking at human protein complexes because the data is way more interesting to look at. And we are looking at an example where one protein is shared between 2 protein complexes. So cap ZA1 is the protein that is shared between the 2 protein complexes. On the left, you got IEX data. On the right, you got your SEC data. Cap ZA1 is in the thicker line, and it has a wide protein abundance profiles, as you can see. And that's a telltale sign that the protein is shared between multiple complexes. And then we did peak picking. And for cap ZA1, we found 5 peaks. So the peaks is pretty complicated. So I simplified it a bit by finding the best pair of peak for every pair of protein. And so it's a lot more simple, as you can see. For the IEX data set, I found two sets of peaks representing two sets of protein complexes, which contain cap EZA1. Again, the thicker line is cap EZA1. For the SEC data, you can see that there's two protein complexes because the one on the top, the complexes at the top, is more shifted to the right, and the complex at the bottom, the peaks are more shifted to the left. But it's not as distinct as IEX, so I think using IEX definitely has its benefits. So what if we combine the IEX and SEC results? Can we get a better score? So we can combine two p-values to get a combined p-score using chi-square distribution, and this is what it looks like. We have p-value 1 on the bottom, p-value 2 on the y-axis, and the color gradient represents the combined p-scores. So what it means is that if you combine two p-scores that are not passing the threshold, then it can give you a score that better passes the threshold. The dotted line is equals to 0.05 for the combined p-value, and anything below the dotted line is the combined p-score, which is better than 0.05. So this is the result from combining IEX and SEC. You have seen most of this graph before, but I've included a new panel which have the p-scores after you combine the IEX and SEC data. We can see that combining the p-scores from IEX and SEC increased the number of significant complexes by about 30% if we use Spearman correlation as a baseline. And again, we draw a Venn diagram comparing before and after combining the p-scores. We find that if we combine SEC and IEX, we found 7 additional complexes, but there's a lot of overlap between before and after combining the scores. But if you combine the score, it drops out 5 protein complexes that were found previously with only either SEC only or IEX only. So in the future, I'll be using machine learning approaches such as random forest to predict protein complexes. I'll use multiple correlation metrics as features, and I'll bring in more extra data like that are orthogonal and give me more biological evidence to do the prediction. And in conclusion, randomization is an effective means to benchmark high confidence protein complexes in PCP datasets. It's great for studying known complexes. And we can assess the capacity by which different features of PCP profiles can be used to identify complexes to tell us, what is the best practice for predicting complexes. Some correlation metrics, for example, Euclidean distance, did not work well in the context of PCP, so we have to be careful what features we use. IEX and SEC identify different complexes, so there's benefits in combining both approaches. And when there are proteins with very complicated peak structure, peak picking can help us identify which peaks is more important and is likely to be associated with the complex. And I would like to thank people like Michele, who shared some inside information on PCP analysis. Jane is funded by our ARC DECRA Fellowship. Alicia for sharing the iHeatMap heatmap package. And I've used HPC, high performance computing from UNSW, and in-house in the Robert Shorty Center, and funding from Bioplatforms Australia and the New South Wales government. Thank you, everyone.
Thank you. Are there any questions in the audience? Oh, let me just pass that one to you.
Thank you.
And then I'll make my way to the back.
I just wanted to make more of a comment than a question. You talked about the very low overlap in papers in Nature and Science on the protein complexes. I just wanted to make the comment there's been a growing realization of reproducibility crisis, and it seems to be occurring in Nature and Science more than any other journal, which are rapidly growing reputations for having the most retractions of any scientific journals. So perhaps it's not quite so surprising that those overlaps are so small.
Yeah, I agree with your sentiment, but they are always the first one on the field, so you never really know what's real or not until someone look at it. So, but they do release their data, and I've used the data that has been released in the public extensively. So that is a great plus for me because I can now benchmark my data against theirs, and I think that is the most important thing, to be able to share data, share the code, share information publicly, and to have them reproducible. Even though you might not get the same score because they're doing false predictions, you can still, improve over time, and that's the great thing about science. Thank you. That's a great comment.
Thanks, that was super interesting. You said that you were working on yeast as a model organism, and then switched to human examples later on.
Yeah.
Is there like a clean transference of the techniques between one and the other, or like how?
Very clean.
Yeah, nice. Very
clean. It's basically the same pipeline. I just changed the data set.
And there's no like variance in the different species or the expression of the proteins that means that the techniques wouldn't work? No,
it's pretty much transferable. So you could basically dump in any correlation metrics and any correlation, put in PCP data, and you can benchmark it against any groups of any kind.
Right.
So
there's no risk that like one method will work for yeast but not for humans.
Yeah, I think if anything, if there's any reason why it wouldn't work, it would be me. So
It's hard to eliminate that.
yeah, hard to eliminate the person, the user error.
Was one?
Sorry, hand go up.
Is that one?
No, no, you're not.
No.
Hand? No.
I had a comment, which is just that I think it's really nice that you're thanking the computer. What we do is computing, and we never really think about what makes that possible. So it's nice that you're thanking the computer.
Yeah, I'm really thanking the computer. I did spend endless hours on it and trying to crash it and burn it. But it's really the funding for the computer wasn't funded by me. They were funded by the government, so they're part of the infrastructure grants. So yeah, I have to thank them. The name is Katana and Clive. Clive is a private one, so it's our own. Can't use it, sorry.
Beautiful. Did you have any intuition why the mutual information metrics didn't work as well as the correlations? I find that surprising.
Oh, that's a great question. There's two reasons. One reason that people have been saying is that it suffers from low power. So if you have very few data points, I've only got 40, 70 data points, then it might actually not be strong enough to detect complex associations. But there's also one thing that I haven't tested. You might like to test it. Is that mutual information only tells you whether it's correlated, but it doesn't tell you whether it's anti-correlated or like the direction of the correlations. So that basically halves the amount of power already. So that could be why. But there are other reasons as well which I haven't explored. Maybe I didn't tune it properly. There are some other tuning parameters that you can use to make it work better, but I doubt it is the tuning parameter. I think it's the power and the fact that it's still very early days for those MIC metrics. Yeah, Loic has helped me a lot with the dealing with the MIC and something called the RIC, randomized information coefficient, but I haven't shown the data for RIC though. Yeah.
Thanks. There are no further questions. Please join me in thanking Iggy once again for this great
Thank
talk.
you.
Thank you.
Thank you.
And it's time for announcements. Is anyone hiring fun new projects? Greg, maybe you?
Hi, Greg. Hi, Greg Pearl from World Recruitment. I want to thank Troy and Josh tonight for videoing both presentations. They should be up within the week on the Data Science Sydney Meetup YouTube site. Please subscribe when you go to that. Eugene would be very happy if you did that. From my point of view, I purely focus on data science and data recruitment. If anyone's got any questions, yeah, look, there's some great companies, everything from startups right through to the larger organisations that are always looking for good people. So yeah, please come and have a chat and link up with me on LinkedIn. Thank you.
Thank you. Anyone else got an announcement to make? Yes.
Bit random, but if any of you are bioinformaticians, then I'd be very interested to talk to you. That's all.
Too easy. And okay.
I am.